Whenever we talk about a new drug, it's not something that suddenly appears in the market. It actually goes through a long structured scientific process before reaching the patient. So, what exactly drug development? It is a complete process of discovering, testing, approving a drug to ensure that it is both safe and effective for use in humans. This entire journey is broadly divided into four major stages, like stages from drug discovery, then preclinical testing, followed by clinical trials in humans, and finally the post marketing surveillance after approval. So let's understand how this journey actually happens. In the early stages, like the discovery plus preclinical, the beginning, scientists screen thousands of compounds. These can be either newly synthesized or already existing molecules to identify something that shows potential through therapeutic effect. So, once a promising compound is found, it is tested in different animal models. This helps researchers understand how the drug behaves in a living system, its safety, toxicity, and basic effectiveness. At this stage, we call it as a lead compound. Initially, there is a very limited data, so it is called an early lead. As more studies are performed and more evidence is controlled, we call it, uh, it becomes late lead, which is stronger and more reliable. Once after this stage, when we have enough and safety, means enough safety and biological data, the drug is considered ready to move into human testing. So there the clinical trials start. So once the preclinical testing is done, the drug enter into the clinical trial phases, which are conducted in phases like phase 1 clinical trial, phase 2 clinical trial, and where the drug is given to the patients who actually have the disease. Here we actually evaluates its effectiveness and continue the monitoring. After that, phase 3 is conducted. In phase 3, this will be on a larger population, and this phase confirm the drug effectiveness, compare it with existing treatments, and identify any rare side effect. If the result from these phases are satisfactory, all the data is compiled and submitted to regulatory authorities, which is NDA or PLA, for the approval. After this successful clinical trial, the company submit an application like NDA PLA to regulatory authorities. And then comes a review period where experts carefully analyze all the data to ensure the drug is safe and beneficial. If approved, the drug is finally launched into the market for public use. And then comes post submission and then this phase 4 post marketing surveillance study. But the process doesn't stop here. Even after approval, phase 4 or this post marketing surveillance continue. This help detect long term or rare side effect and ensure ongoing safety. So from discovery to patient use, drug development is a long, rigorous process designed to protect human health. Now, if we'll go stage by stages, the stage 1, drug discovery. This is where everything begins. Scientists are looking for a new molecule that could become a medicine. This is where the three approaches you mentioned, like pharmacological, toxicological, and chemical, these three approaches are there. And scientists select a lead compound from thousands of possibilities that shows the most promise in hitting the disease target. And these three main approaches are pharmacological approach. This approach, here the scientists identify a specific biological target. So this could be an enzyme, a receptor, an ion channel, or a protein that play a role in a disease. So that's why we call it rational drug design or target to lead. This could be anything. Then they search for a molecule that can interact with this target in a useful way, either blocking it or activating it. For example, in hypertension, scientists knew that ACE enzyme was involved in raising blood pressure. So they looked for a molecule that could inhibit ACE, and that is how ACE inhibitors were discovered. Next approach comes to toxicological. This might seem strange, but some of our most powerful medicine came from studying toxic substances. So scientists take a toxic compound and think, can we modify this to make it useful in medicine at a controlled dose? So some early chemotherapy drugs also came from this toxicological approach only, like from studying nitrogen mustard. a chemical warfare agent. Scientists found it killed rapidly dividing cells and used that property to target cancer. Then the third and the final approach is chemical approach, also called the structural modification, where we study the structural activity relationship. Here the scientists take an existing drug molecules that is already working and modify its chemical structure to make it more potent. So it also which can improve the bioavailability, selectivity, and extend the half-life. So it will be more potent, more selective, and have fewer side effects. For example, like modifying penicillin structure gave us amoxicillin, broader spectrum, and better absorb. Next, the second stage comes preclinical testing. So once a promising molecule is found out, it cannot go directly into humans. So first it must pass preclinical testing. This happens in two ways, in vitro and in vivo. In vitro means testing in the laboratory. Does this molecule actually do what we think it does? Does it hit the right target? Next is in vivo. In vivo means testing in the animal. So the molecule is given to animal, usually mice, rat, and then larger animals. And scientists study pharmacokinetic, how the drug is absorbed, distributed, metabolized, excreted. Then the toxicity. They with ADME they also study pharmacodynamics, what the drug does to the body, and most importantly, they study toxicity. Is this drug safe? What dose cause harm? What are the side effects? So all these things will come in preclinical testing. Now next comes IND applications. Now the company has a promising molecule. Preclinical data looks good. Now they want to test in humans. But they cannot just go ahead. They must first file an IND application. This is investigational new drug application with the regulatory authorities. In India, this goes to CDSCO, like Central Standard Control Organization. In many countries, like USA, it goes to USFDA. So what does the IND application contain? It contains three main components. First, where the all preclinical data, like every animal study, every toxicity result, every pharmacokinetic finding must be submitted. Second, the chemistry, manufacturing and control data, like CMC. This tell the regulator how the drug is manufactured, what the drug composition, how it is tested for quality. Third will be the clinical protocol. This is a detailed plan for how human trials will be conducted, who will be studied, what doses, how safety will be monitored. Once the IND is filed, the regulatory authority has 30 days to review it. If they do not object within 30 days, the company can begin phase 1 clinical trial. If the regulator has any concern, they issue a clinical hold and trials cannot begin. So this IND step is a legal permission to move from animal testing to human testing, and it is a critical gateway in the entire process. Now next comes, after this only clinical trials start. So once the clinical trial is going on, with this, once the IND is being processed, the drug characterization happens alongside and after preclinical testing. So this is where scientists study the drug molecule in complete detail. What exactly is studied in drug characterization is first physicochemical properties. What is the molecular weight? Solubility. What is pH stability? Does the drug break down in heat or light or moisture? What is the pK? This affects absorption in the body. Next is pharmacokinetic characterization. How is the drug absorbed? What is the bioavailability? How is it is distributed? It is a CYP 450 substrate. How is it excreted? And then this analytical validation is also there. So, which help to develop the precise method to measure the drug concentration in blood and tissue. So, all these characterization data is essential for designing safe clinical trials and eventually for writing the prescribing information that goes with every medicine. Next, doses form development. Now, this is the stage where it is directly related to the pharmacy knowledge. A drug molecule by itself is not a medicine. So you cannot give a patient a raw chemical powder. So the drug must be formulated in a doses form. A tablet, capsule, injection, cream, inhaler, whatever is most appropriate. So doses form development ask several important questions. What route of administration is best for this drug? If the drug is destroyed by stomach acid, Oral route may not work. Maybe injection is better. If it needs to act locally in the lungs, inhalation is ideal. So what excipients should be there? Binders, filler, disintegrants, preservatives, all must be selected carefully. Will they interact with the drug molecule? Will they affect stability? What is the target release profile? Should the drug release immediately for a quick effect or like a painkiller, or should it release slowly over 12 to 24 hours? Formulation. Scientists develop and test for multiple formulations before selecting the best one, and this optimized doses form then goes into the clinical trial. Then the clinical trials where phase 1, phase 2, which will be phase 1 will be in 20 to 100 healthy volunteers, phase 2 will be 100 to 50, 500 patients, phase 3, 1000 to 5000 patients. This will continue. Next comes regulatory review. Once phase 1, phase 2, and phase 3 is complete, the company submit a new drug applications or biologics license applications, which is called BLA. So FDA regulatory review, a team of experts like physicians, pharmacologists, statisticians examine all the data, developing the specific instructions like warning, uses guidelines for the drug. Approval, if the benefit outweigh the risk, the drug is approved for the market. And last, the final comes post-marketing safety monitoring. This process doesn't end at approval. Regulatory bodies continue to monitor the drug once it is available to the general public. Tracking rare or long-term side effects may not have appeared in earlier trials, which can come later. So routine check of manufacturing facilities to ensure consistent quality and safety protocols. So this is how all the stages in drug development process. So if we'll quickly recap or the summarize, stage 1, drug discovery, 3 approaches, pharmacological, toxicological, and the last one was chemical. Stage 2, preclinical testing, in vitro, in vivo studies, pharmacokinetic, dynamic, and toxicology. Stage 3, IND applications. Stage 4, drug characterization, then dosage form development, then this post-marketing surveillance. So this entire process takes 10 to 15 years, and out of thousands of molecules, only one or two become actual medicine. So this is why new drugs are expensive and why clinical research is so important. If you found this video helpful, please subscribe.